Talk 3 is a master infrastructure setup for multiple pipeline integrations in minimal residual disease. There are 3 speakers, and this is a flagship talk. I think it's 60 minutes or so. Speakers are from both Myriad and Strand. Speaker 1 is Benjamin Weishville, staff software engineer at Myriad. He brings over 20 years of software engineering and bioinformatics experience, holds a master's degree in computer science. Previously, he was one of the creators of Ergo 2.0 at EigenBio, where he was principal software engineer. He's worked closely to develop bioinformatics solutions for NIH, Cargill, Pfizer, the USDA, and others. At Myriad, he's on the team creating the infrastructure for the MRD assay, focusing on the data management system, orchestrator, and analysis manager. Speaker 2 is Kirti Molika, software engineer at Strand Life Sciences. Kirti has been contributing for the past 4 years at Strand, is a 2021 graduate of IIT Roorkee. She specializes in back-end development while also working across the full stack. Experience in large-scale software projects, she focuses on designing and delivering scalable, reliable systems, strong interest in problem solving. Speaker 3 is Eric Anderson, staff software engineer at Myriad. He's worked here for 11 years with over 2 decades of experience spanning video games, digital signage, planetaria, health records, and photo book publishing. He's been a key contributor to several major architectural shifts at Myriad, including test data frameworks and an automated regulatory documentation system. He's currently co-architect of the company's data management system. Over to all of you.
Today, I'm going to be focusing on, we're going to be focusing on the engineering questions, and we're not part of the scientific team, so please keep that in mind with any questions. So for the agenda today, I'm going to start with an overview of MRD and the infrastructure, and also zoom in on a couple of problems that we encountered while scaling up our infrastructure. Then that'll be followed by tumor profiling pipeline deep dive by Molika from Strand Life Sciences, and an overview of the data management system by Eric Anderson from Myriad Genetics. And then at the very end, we'll have a Q&A, so you can follow. If you hold all questions to the very end, that would be very helpful. Thank you. The precise MRD test for Myriad Oncology is an ultra-sensitive tumor-informed assay that utilizes whole genome sequencing. Molecular residual disease, or MRD, refers to the small number of tumor cells that remain in the body during or after treatment. These tumor cells shed DNA into the bloodstream, which is known as circulating tumor DNA, or ctDNA. Our assay can detect the presence and quantity of ctDNA in the plasma of cancer patients with residual disease or tumor recurrence with a high level of sensitivity, with tumor fractions as low as 0.0001% or 1 part per million. This is done by using up to 1,000 carefully selected and personalized somatic variant targets that were identified during tumor profiling. This test can be used for monitoring ctDNA levels throughout a patient's clinical cancer care, providing the patient with answers to 2 primary questions. Is the cancer treatment working? Has the cancer recurred? Unique features that Myriad brings to MRD are the comprehensive and personalized whole genome sequencing tailored to a patient's individual tumor, up to 1,000 variants that are used to build the panel for circulating tumor DNA detection. This offers us a few advantages compared to other MRD tests. Precise MRD is more accurate when the tumor is smaller, and not as much of the initial tumor sample is required to run this test. The assay supports multiple indications, including those such as breast cancer, which are low shedding. It can precisely quantify how much ctDNA is present, which is very important in determining whether a treatment is working. So earlier detection leads to higher chance of intervention. So if we were able to detect at a lower time, we were able to, treatments can be applied earlier, more likely to save somebody's life. MRD is a complex assay, but in summary, a tumor sample and the buffy coat are isolated from whole blood. The buffy coat is isolated from whole blood and are sequenced on the NovaSeq X. This sequencing is used to call somatic variants present in the tumor genome, but are not present in the germline genome, coming up with detection probes. These variants create that detection probes for targeted sequencing of the patient's plasma and other subsequent time points. These are also sequenced in NovaSeq X. So this starts with the baseline and adds these additional time points leading to the patient results. MRD presents a suite of new technical challenges. The scale of the data is much greater than we have encountered in the past. Sample data now expands from 1 to 3 gigabytes per sample to 300 gigabytes per sample, now with multiple time points for each patient and multiple samples for each patient. The orchestration is much more complex. Multiple samples for each patient means that data availability patterns are much different than before. The orchestration of getting the data where it needs to be for analysis ends up being quite complex. We also need to make sure that we are also keeping in mind integrity and compliance. Shuttling data between lab and 3 cloud systems introduces challenges, ensuring data integrity at every point, and that data provenance must always be demonstrated. Now I'm going to talk a little bit about the infrastructure. When you start in R&D, you need to focus on agility. You need to be able to iterate quickly. It just needs to work. This leads to temporary patches, and those temporary patches can lead to instability, mistakes, and interruptions. These are expected since we're moving fast. Building on top of existing systems is normal, but those existing systems may not be a perfect fit for your technology. And so these systems tend to accrete technical debts because of this. Performance is unpredictable, but that's okay. The inputs and outputs are also unpredictable. We're not really sure what to expect out of it during R&D. However, in production, the needs are much different. We're now building for long-term use. This needs to be engineered rock solid. We need to be able to have data provenance and integrity tracked at every step. The input, however, is now predictable, which gives us some advantages, which make it easier to make performance much more predictable. Now systems must be modular, understandable, so they can be supported and extended far into the future. This is so what does this bring us to? Brings us to our production infrastructure, this diagram. There's a lot of parts of this diagram, so I'm going to go through it piece by piece. First, we're going to start with the Myriad lab, which houses the Illumina NovaSeq X sequencers. We have AWS, which houses our data management system, our S3 data stores, and our orchestrator. The orchestrator, which is implemented as several state machines in AWS step functions. This will be covered in greater detail in Malika's talk. We have the data management system, which is a series of lambdas connected to API gateway and event bridge. This will be covered in detail in Eric's talk. Illumina Connected Analytics, or ICA, which is connected to our S3 bucket and many S3 buckets, and performs demultiplexing and DRAGEN alignment with a custom pipeline. DNAnexus is where the primary bioinformatics analysis, such as tumor profiling and ctDNA detection, are run. We send our probes, probe orders to a DNA synthesis company, and then we have a custom LIMS system as well, all of this in effort to bring a report to the patient. So I'm going to run through what the data flow looks like for a sample. So for WGS samples, we're talking about tumor, buffy, and controls. So not the targeted sequencing, but in doing the tumor profiling. The sequence data, after it's been sequenced, NovaSeq X, the data is uploaded to ICA, which is connected to our S3 bucket. Dropping those files in the S3 bucket cause S3 events to propagate to an event bridge. More of this will be covered later. Those get registered to our data management system. And at the same time, these also get registered with DNAnexus on storage 2.0. So those can be seen inside of a DNAnexus project. Next, our orchestrator takes control. The first analysis is demultiplexing in ICA. So this is recovering the individual samples from the flow cell where the samples were combined. Orchestrator starts and monitors the job on ICA utilizing Illumina's BCL convert software, and this updates the LIMS and the data management system as that process goes on. After the results are reviewed, excuse me, I missed one step. Next, our orchestrator executes and monitors a DRAGEN alignment job, also on ICA, and this also updates the LIMS and DMS. Those results are then sent to the LIMS where somebody reviews them, and if they look good, and we have a buffer and tumor sample that pass review. We can then proceed to tumor profiling. Tumor profiling is initiated by the orchestrator by checking a queue in the LIMS. This orchestrator then moves the alignment data, initiates the workflow, execution in DNAnexus, and it tracks the files. It creates and updates in DNMS. After the execution completes, the results are then returned to the LIMS via the orchestrator. LIMS then initiates order for the probes. At the probe synthesis company, those probes are returned. The probe details are returned to the LIMS. And then when detection is run, this process is very similar. However, one of the inputs now is going to be the probes for targeted alignment. How can we accomplish this? So this infrastructure is implemented with modularity at multiple levels. First is some microservices. We decoupled services such as our LIMS and orchestrator. While R&D, in R&D, this tight coupling made sense as the LIMS, the orchestrator, and the assay were developed in tandem to support the scientific needs. However, this no longer makes sense in a production assay. We've moved towards a solid contract between systems and placed a service in front of the orchestrator to abstract the inner workings of the orchestrator out of the LIMS. Workflows are composed of reusable WDL modules. When workflows into development, it was common to copy and paste between repository sections of a workflow. However, in production, this system has been replaced with published artifacts that can be referenced in a master workflow. A versioning service ensures compatibility between the modules of the assay. As the assay moved to production, there emerged a need to track compatibility between different modules, their inputs, and others between the two parts of the assay. This grew into a versioning service, which ensures that only compatible versions of detection and tumor profiling are used together. An additional service called the MRD translator helps maintain compatibility between these versions as well. Now I'm going to focus on two specific problems we encountered when scaling up our software systems for MRD. First in our orchestrator, and then one in the DMS. So recall that I mentioned that our orchestrator is made up of several state machines. A few operations in our state machines run per sample need to handle resources such as deleting temporary files or creating projects for the entire batch. These operations aren't necessarily slow, but they do take an unpredictable amount of time just due to random network latency. These lambdas run with concurrency of 1, meaning that only one execution of the lambda is allowed in the account, while an exponential backoff worked for a lower number of simultaneous state machine executions. As the number of executions increased, so did that likelihood of those executions ending up backing off at the exact same moment. So if I can illustrate this. So this is a square wave function where the wait times are illustrated in the valleys, the peaks being execution attempts. If everyone attempts the execution at the same time, and the more of them that you're attempting at the same time, the more likely this is to happen, then you give them all an exponential backoff. But they all receive the same exponential backoff, then they will all try to execute at the exact same time again. We only encounter this problem around 80 simultaneous executions. That could have just been unluckiness. It could easily that we would have encountered it at a lower number even. So what were the possible solutions that we considered? We could limit how many state machine executions could happen simultaneously. Well, that might have worked okay and made it less likely that we'd encounter this problem. It would still could have happened just out of pure bad luck at any time. Excuse me. We could move these problem tasks. So why even do these operations here inside of the state machine, right? So we can move them out. And this is definitely something we're considering for a future solution. The other possible solution was to introduce a small jitter. So we prevent too many of these simultaneous events. So before, where execution attempts were synchronized and the backoffs are synchronized, by introducing a small jitter during every backoff attempt, we desynchronize those next execution attempts. So they no longer overlap. And now we can scale over 5 times from that 80 to over 500 simultaneous state machine executions, though, to be honest, we haven't actually seen the limit of how high this could go. Probably up to the account concurrency limit. Speaking of which, that is the next thing I'm going to talk about. So the next concurrency issue was exactly that. Too many events overloaded the lambda concurrency limit of an AWS account. So every AWS account has a certain number of concurrent executions that are allowed. And I'm going to go over briefly just a top level of how the DMS is set up because it's necessary to understand this, but we're going to go over it in more detail in Eric's talk following mine. DMS is set up to listen events from buckets, from other accounts, from other regions, and these are all fed into EventBridge. Our first naive setup of this had everything feed every event that hit EventBridge led to Lambda execution, which then led to adding something to the database. As you might imagine, this immediately came into some problems. First off, our dev databases didn't allow too many concurrent connections, so we quickly exhausted how many connections that our database allowed, and had Lambda failures at that point. So we added a proxy. So with serverless, you don't really get a connection pool like you would on a big server, and you can just have set up your database with a large connection pool. But with an RDS proxy, you get basically that, a connection pool. And this worked great. Now we could scale from the few dozen when it broke before to hundreds or even thousands of requests in a minute. However, now a new problem emerged. As we got fed, say, 1,000 events in a second, this quickly consumed all the Lambda concurrency in our total account. So this only goes up to 817 because we have a reserved 200 for certain tasks that are not part of this count. But as we hit this total level here, this 817, that means other Lambdas, other state machines can't execute. They get a too many Lambda request, I think is the error. So now what can we do about this? We re-architected things a little bit. Instead of having the event bridge immediately execute this Lambda, we now go to an SQS queue, and then these events are batched and then sent to the Lambda in groups of 10. Now that Lambda is still connected to the RDS proxy, so we still have great connection pooling to add it to our database. And in addition to this, we also added a concurrent execution limit of 20 on this Lambda. This easily scaled to thousands of events in a second without having any issues whatsoever. It was definitely a very interesting problem to solve at the time that it happened. I woke up with that unfortunate news of using all the Lambda concurrency. on the account. So hopefully our information will prevent that from happening to you. Our next talk is a tumor profiling pipeline deep dive by Malika Kurti from Strand Life Sciences. Thank you so much.
Hi everyone, this is Malika, and I'm going to continue on what Benjamin was talking about. Today, I'll be walking you through a journey of building and automating this whole MRD WGS alignment pipeline at Myriad, that is the whole genome sequencing pipeline. We'll take a look at the whole complete process overview, the challenge which was faced, the solution we built, the impact it actually had on cost, scalability, and automation. I'll also give a little deeper look into the architecture and two of the main workflows which are being used here, that is demultiplexing and DRAGEN alignment, and how we actually implemented them using the AWS step functions so that we can ensure reliability. So I was talking about taking a closer look at the journey of data from the moment we have the patient samples to the point we actually get usable genomic information. So when we have the samples of flow cells, we actually initially load them into a sequencer, right? And the sequencer, it then reads and produces what is called as BCL files. The BCL files are actually the raw machine outputs. They are mostly Illumina specific and they aren't very useful on their own. They are basically like sensor reading straight from the machine, right? So the first step would be to actually run what is called as demultiplexing on them. This actually runs demux and it converts the BCL files into what is called as FASTQ files. The FASTQ files are more universal and organized format of files. It consolidates all the sequencing runs into a format that can be used by any downstream analysis. So let us initially here note that the BCL files are actually outputs of the entire flow cell, which is stored across multiple files, whereas the FASTQ files are per sample files. And they basically have two files, read one and read two. So once we have the FASTQ files, we run an alignment step. So what does this alignment do? It actually takes all these reads and they map them against a reference genome so we can exactly see where each step belongs. Finally, the results of the alignment are stored in a format which is called as a CRAM. This is the version we keep for any downstream analysis of, suppose, the variant calling, MRD detection, or tumor profiling, etc. So originally, the whole process implementation, it had two major problems. The first one was it was too slow, and the second one, it was too expensive. It was too slow because the whole process, it actually relied on CPUs based on DNAnexus. So the process, it was too slow and it was too expensive. The process was very slow because the whole process, it relied on CPUs based on DNAnexus, and the BCL to FASTQ conversion and the BWMM, it took a very large amount of time. And it was too expensive because each whole genome sample, it was costing a lot. And the reasons might be varying. It might be because of the instance type we were using in DNAnexus or the amount of time the instance types actually run for. So what was the solution for it? The solution was to move to a platform that actually uses a more specialized design for this task, that was doing DRAGEN alignment on ICA. And what was the result of this? It actually resulted in a 90% cost reduction per genome sample, and it actually made the process relatively faster. So with this, we were able to move from a costly to relatively cost-efficient process while automating all these steps. So now let us take a look at how the workflow is actually currently set up today. There are three main components that are being used. One is BaseSpace, the other is ICO, which is Illumina Connected Analytics, and the other one is DNAnexus. So it initially starts with the BaseSpace. This is a system which is connected to the sequencer. You can think of it as a place where the raw data initially lands once the sequencing is actually complete. From there, the data is pushed into ICA. ICA and BaseSpace are actually connected to each other through a project, and ICA is also a platform which can be connected and backed up by S3. So once the data is pushed from BaseSpace to ICA, this is where all the heavy lifting happens, like all the alignments or analysis are run here. So basically the raw data, it is being converted, it is being cleaned and aligned in ICA. So you can think of ICA as the main processing engine here. So finally, once when we have the results, and the results are stored in DNAnexus. DNAnexus is basically used for our long-term storage and analysis environment. It is where the alignment data lives and where any other tools can pick that alignment data for any further analysis. So we have tried automating this entire journey, like from the base space to DNAnexus, so that people don't need to watch over every step of it using what we call as AWS state machines. So how exactly did we do it? We designed what is called this MRD WGS alignment pipeline, whose purpose is very simple. It is to take the raw sequencing BCL data, process them, and to make sure that the results end up in the right place for any further use. So behind the scenes, the pipeline actually uses two automated workflows, which are demultiplexing and DRAGEN alignment, which are implemented using state machines. So now let us take a look at each of them. Yeah. Before moving on to look on what these workflows exactly do, let us take a look at what actually are these step functions and the state machines that I have been mentioning till now. So at its core, these AWS step functions, they are actually a way to orchestrate a multi-step process. You can basically think of them like a flow chart, and that doesn't only show the steps, but they actually runs them for us. And each step know what to do, what to check for before starting, and what to do if actually something goes wrong or something fails. So inside this step function, we define what are called as the state machines. The state machines is basically a blueprint of the complete workflow. It describes the order of the task, how each decision is made, and how the errors are actually handled. Why are we using this? Because genomic data processing isn't a one single step, right? It includes a series of checks, conversions, analysis, transfers, etc. And with step functions, we can actually trigger the right step at the right time. We can even visually monitor the process in real time. And we can let the system handle all the retries, delays, or way to handle failures without any manual intervention. So these step functions are what makes a pipeline a little more automated, a little more robust and transparent. We have implemented these state machines using a Python package, which is called as AWS Troposphere. So now let us first look at the demultiplexing state machine. So this state machine is actually designed to automate the whole demultiplexing process. That means going from the BCL data, which is produced by the sequencer, all the way to generating the FASTQ files and any associated quality metrics. So in this process, the two main systems being used are BaseSpace and ICU, which is Illumina Connected Analytics. So the BaseSpace is used to check if the sequencing run is actually completed and that the raw data is available for us and we can fetch it. And the ICA is used because this is where the actual processing happens, right? This is where we are trying to do the demultiplexing. And it is also used because of its cost and licensing consideration. It also has a flexibility to build any custom pipelines, and it also has ability to retry or requeue a particular sample. So how does this actually work in practice? So I'll be showing you a simple workflow of what actually happens inside this demultiplexing state machine. Yeah. I do want to make a note that each of the box which is mentioned here is actually a state inside the AWS state machine. And here a state, it can actually represent a task, a choice, waiting, polling, or any kind of action. So initially, the system, it checks for the FASTQ data. The system looks for the FASTQ data because if the files are already present, we don't want to duplicate the work again and do the whole process again. So if the files are already present, we say that the data exists and we finalize the output. But if the files are actually not present, then the system, it moves on to the next step, which is to verify if the sequencing is completed. So this step actually polls BaseSpace to check if the sequencing run is fully complete and the data is actually uploaded correctly. Since the sequencing might be in progress, we poll this base space for every few minutes to check if it has been finished. Once the sequencing is finished, we try to find the sequencing director through the run ID. And this is being done in ICA because the base pairs in ICA are linked to a project, as I've already mentioned, right? So once we find the sequencing directory, we start a health check analysis. It is to see if all the files are present and that no required file is actually missing out from the folder. The health check pipeline is actually a custom pipeline that Strand has helped port into ICA. Then we poll for that health check analysis to complete and then start what is called as the BCL convert. That is the demultiplexing step. This demultiplexing converts the BCL files into the FASTQ files and where poll for this analysis to complete. And then finally, when we have the FASTQ files as a result, we tag those FASTQ file folders so that any downstream tools or analysis can actually find them and use them very easily. After which we say that these are the outputs and these are the places that are presented, and we finalize that output. So all the steps that I have mentioned, they run automatically one after the other. Like if something goes wrong, for suppose if a step actually fails, the system, which is state machine here, it is configured to retry or flag any error appropriately. Let me give you a small example here. For suppose while finding a sequencing directory, it can go in multiple ways, right? You may find a sequencing directory, you may not find it, or you may find multiple folders or any other kinds of error. So the state machine can be configured in different ways. Like if you did not find a directory, we can say it. We can configure the state machine so that it actually fails. If you find multiple folders, it means that somewhere something went wrong. Even in that case, we can say that we can configure it so that it fails. And for any other errors, we can also do it such that the whole process that is finding the directory, it can be retried up to a certain limit. And the limit can be around 3 or 6 as per our convention. So once the demultiplexing is actually complete, we have our FASTQ files. So after having a FASTQ files, we now move on to the second workflow, which is the alignment. So now let us talk about the DRAGEN alignment state machine. This state machine is designed to automate the whole DRAGEN alignment process. That means taking the FASTQ files that was produced from the previous state machine and then aligning them against a reference genome, which ultimately gives us the cramp files and metrics. So in this process, the two main systems here used are ICA and DNAnexus. ICA is the place where the alignment actually runs. It takes the FASTQs and uses the DRAGEN alignment to perform this cost-efficient alignment. And then the DNAnexus is where we are actually going to store our final results. So now let us take a deep look into how this process is actually implemented inside the DRAGEN alignment state machine. So what happens here is the system at first check if the FASTQ data for that particular run is available or not. So if it is not available, it means that data actually doesn't exist and we have to run the initial demulk state machine first. So in that case, we say that we did not find the data and we just failed the state machine. But in case you have found the FASTQ data, we move on to this next step, which is to creating projects in ICA and DNAnexus. So this ensures that we actually have a place to run the alignment, that is ICA project, and a place to actually store the results, that is the DNAnexus project. So if these projects are already available, we skip creating them. But if they are not, we have made it such that they actually create the projects automatically. So after we are done with these two steps, we start the DRAGEN alignment pipeline. So this DRAGEN alignment analysis is launched on the FASTQ files, and this analysis is also polled until its completion. So once we know that the alignment is actually finished and completed, we symlink the AWS S3 data to DNAnexus. Here I'm mentioning AWS S3 because the ICA project here, which is being created, is being backed by an S3 bucket. And symlinking is actually a new feature which is put up by DNAnexus. It enables us to link any external data on S3 or any other such places and store them as objects on the DNAnexus platform. So this is what is the DX storage 2.0 that even Benjamin has been mentioning. So after symlinking these AWS S3 files to DNAnexus, there is something called DMS injunction via DMS injunction of data via an event. And this DMS here is a data management system which is going to be covered by Eric. And this is a system which is being internally designed by Myriad that handles all the tracking of files and any other data management. So now we have our CRAM files, that is the results of the DRAGEN alignment, right? So those files are now tagged so that they can be used by any further downstream tools or analysis and can be easy to locate. And then we finalize these outputs and give them as results. So once we have the CRAM files on both the state machines are run, and the complete data is shifted from AWS S3 to DNAnexus for our long-term usage. From there, this data can be further used in other analysis like tumor profiling or ctDNA workflows or somatic calling, etc. So what has this transformation actually achieved for us? We have moved away from a very expensive CPU-based setup to a much more accelerated system by using the DRAGEN alignment in ICA, which is comparatively faster and relatively more efficient. And the result was also a major cost deduction that is almost 90% per genome sample. And I think that is a very drastic saving, especially when we try to scale up and process more and more of these genomes. Apart from this cost and speed reduction, there are other major important benefits also. One is that we now have a scalable and a reproducible architecture. And this is not just one-off solution. This system, it can grow with new features coming up. For suppose the symlinking that I have mentioned is actually a recent feature put up by DNAnexus, and that can be integrated into the present state machines. And the other one is we have achieved a degree of automation. So the workflows which I have mentioned, they run on their own. They automatically handle the polling, the retries, any error recovery based on the rules we have put inside the state machine. That means our team doesn't need to spend a lot of time babysitting the whole process. So to wrap up, I would like to say that this transformation on moving on from complete DNAnexus setup to using all the other platforms like ICA has also has enabled us to give an MRD WGS pipeline that is way faster, cheaper, scalable, and reproducible as we are using the state machines for it, and they are automated to a certain degree. Yeah, thank you. That will be all my side, and I'll be passing on to Eric to go on detail about the DMS system.
Thank you, Malika. Gonna go ahead and hijack your share.
Yeah, please do that.
All right. Let me just confirm that this is looking right. Yeah? Okay. Hi, I'm Eric Anderson. I'm a staff engineer at Myriad Genetics. For the past year, I've worked alongside my co-architect, Benjamin Weisville, to design and build Myriad's Analytical Data Management System, or DMS. You've already heard a little about the DMS tonight. This is the whole story. We've learned about the complex orchestration involved in productionizing MRD. This complexity involves or creates three data-related problems: validation overhead, compliance risk, and redundant data. Files flow from platform to platform are used to create new files that in turn also flow from platform to platform and ultimately arrive at their final destinations at many places throughout the system. Before we can use any file that was transferred from one system to another, we need to validate the transfer by comparing the original file's checksum against a new checksum calculated using the destination file. Every orange arrow here indicates a required validation. These validations need to be recorded in an auditable manner. Once we've transferred a file from one platform to another and validated the transfer, we end up with 2 copies of the same data. Once analysis is complete for a sample, many files are no longer needed. All of this is compounded by scale. We expect to handle hundreds of millions of files and petabytes of data across our past, present, and future assays. It turns out addressing all of these concerns across so many platforms with such a complex graph is a bit of a novel problem. We spent months hunting for a vendor offering that could directly handle our needs and came back empty-handed. So we rolled up our sleeves and considered how we think about our analytical data. We thought about how to contextualize our data so we can find it, understand it, and manage it. We wanted to prevent mystery data like here on the left, without a clear origin or purpose. A lonely orphaned file without context isn't just an anomaly, it's also a very real liability. If we don't know a file's story, we can't manage that file without risking operational or compliance hiccups. So to solve these problems, we need to avoid losing context. Every file has a rich network of metadata available at creation, and we use the DMS API to capture it immediately on creation. With this context, we can answer any number of questions about the performance of our system, the cost of our data, the provenance of a given file, that sort of thing. Because redundancy, file integrity, and primary data protection are critical, we think about files a little differently. Traditionally, we're used to thinking of a file as whatever lives at a given path, like we see here on the left. If you copy it, rename it, move it, suddenly you've created a new file. This view fragments our understanding. Across platforms, it becomes impossible to know which copy is definitive, which is derivative, and which to trust if errors appear. The DMS adopts a different model, an identity-based view, where a file's identity comes from its content, not its path. So we see here, a file's identity includes all of the different places it might be copied to. And in this case, we can see that we treat the S3 file location as definitive or primary, where the others are considered copies. This shift in perspective lets us eliminate redundancy by recognizing duplication no matter where it lives. We treat transfers as replications instead of mutations, so integrity can be verified at transfer simply and directly. And we are able to differentiate natively between losing a copy and losing the data itself, a critical distinction for compliance and audit. So how do we achieve this? The DMS is built as 3 coordinated stacks: a database, event, and API. Like the state machines Malika described, the DMS is built using AWS CloudFormation. The RDS stack provides the DMS with its data backing, where all the information about managed data resides, using a relational database. The event stack enables the DMS to do things like perform scheduled operations, respond to changes in a monitored S3 bucket, retry incidental failures, and notify administrators if anything goes awry. EventBridge handles this by feeding events to things like Lambda handlers, SQS queues, or SNS topics. The API stack provides a RESTful API through API Gateway, which handles things like domains, URL routing, and authentication. The API passes authenticated requests to serverless functions called lambdas, which execute the requested operation. This compartmentalization reduces deployment overhead and gives us conceptual boundaries that guide how we think of a given capability. Each stack is provided the appropriate permissions to interact with the other stacks, so events can make API calls, API handlers can trigger events, and either of those can interact directly with our database. We can directly interact with the DMS through the API, either as a user or as part of the state machine-based orchestration system. We can also monitor and manage our data without human interaction via well-defined events as they happen. This is all facilitated by the tools AWS offers to handle common ops concerns like secrets, authentication and authorization, logging, backups, and more using a simple programmable interface. The event-driven features of the DMS are especially exciting, since they transform the system from a static metadata repository to a true data management system. We use EventBridge as our central nervous system. It receives events from other systems and from monitored S3 buckets, or from a configured schedule, and then routes them through event buses to filters that allow us to target behaviors as broadly or narrowly as needed. We use queues to buffer bursts of traffic or events. We use topics to broadcast a single event in parallel across independent domains, such as compliance, cost tracking, or redundancy elimination. Lambda functions do the actual work. They spin up instantly, scale automatically, and vanish when idle. All of these capabilities stitch together to create a dynamic system that not only stores metadata, but actively manages it. Records stay in sync. Policies execute automatically. Anomalies are detected immediately, and both automated and human responses are coordinated in real time. Combined with our API, it gives us enormous flexibility and power to scale against truly large data sets, while remaining confident that each step we take is correct, observable, and reportable. Let's walk through how all this works together in the real world. Malika has already described in detail how our whole genome sequencing pipeline works. How does its data fit into the picture? Let's take a look. So our sequencers spit out the BCL files, and those BCL files are then uploaded to BaseSpace. The BaseSpace BCL file is then used in ICA in the demux operation that creates these fast queues. Those fast queues are then used in the ICA DRAGEN alignment to create CRAM files. The CRAM files are copied to DNAnexus, where they are run through the variant caller to produce VCF files. At each step along this way, the orchestration layer calls the DMS API to register new file locations and their metadata, such as provenance, checksums, the creating process, that sort of thing. Using that metadata, we are able to implicitly determine file identity, which allows us to think of duplicate files, like for instance, these BCL files, or these CRAMs between the DRAGEN ICA space and the DNAnexus variant calling space, and treat them as the same file identity. The redundancy is visible and manageable. Transfers can be validated simply, and definitive files can be preserved as a matter of policy. This allows us lifecycle control via policy. So once the workflow finishes, DMS policy decides what to keep and what to retire. The BCL files are deleted once demux is finished. We don't need them. We treat the FASTQ files as our primary data source. The FASTQ we only need once, and we don't expect to use again except but extremely rarely. So once the workflow is complete, we will compress that FASTQ and shuttle it to our long-term archival space. In the case of the CRAM, we do have this duplication between the ICA space and DNAnexus. We want to retain one of these CRAM files live so that it can be used for additional research going forward. However, the other file can be disposed of safely, and we do. Finally, the VCF file, once it has been used to generate our patient report, is also retained as our final result. And so we, in turn, shuttle that as well into our long-term archival. Now, our archive files are then replicated cross-region into another S3 bucket for disaster recovery and other data resilience features. This way, the DMS not only tracks files, but actively governs their lifecycle. By tying together workflow events, file identities, and storage policies, we ensure data is accurate, its integrity and provenance is always clear, efficient. By eliminating redundancy, we control our costs, and compliant. Each step along the way, we produce auditable, directly relevant records of what was kept, retired, or archived. We have a little extra time, so let's dive deeper into how the DMS thinks of a single file, using CRAMs from the previous slide as an example. To begin with, let's consider a simplified CRAM policy. In this case, we have already mentioned we want to keep an original, a copy of the CRAM file live. So let's keep the original, the one on ICA. As for the duplicate, the copy, let's delete it 7 days after an analysis succeeds. That'll give us a broad grace period that we can use in case we want to do extra work with that CRAM copy. Finally, let's add some resilience to this. Let's notify admins in any sort of anomalous state that isn't expected, or if there's a failure that needs to be repaired. And finally, if we notice that the primary is missing, but the secondary is still available, attempt to repair by copying that secondary back to the primary location and then validating the transfer to make sure that the identity of the file is maintained. Here's how that policy then is expressed in the workflow. After DRAGEN alignment finishes, the orchestrator sends a message containing metadata about the analysis, its input FASTQ, and its output CRAM to the DMS. The DMS recognizes the CRAM and applies the appropriate policy, which in this case is to wait. The orchestrator then copies the CRAM file to DNAnexus and registers the transfer and validations with the DMS, just like it did with the alignment. The DMS expects this copy and in turn does nothing. After copying, the orchestrator runs variant calling and registers its CRAM input and VCF output. The DMS recognizes success. After 7 days, the file is retired and the action recorded. Along the way, if failures or anomalies appear instead, the same event flow triggers the right policy action, perhaps to wait longer, perhaps to alert admins, perhaps to attempt repair, or some other third combination or fourth combination of all of these possibilities. This way, our policies are provided at once and the DMS enforces them consistently across workflows as they execute. As the system changes, the DMS reacts and manages along the way. And that is how the DMS thinks about data. Now, working in bioinformatics, the challenges we're tackling with the DMS aren't new. Provenance, auditability, archival and deletion have always been part of the job. What is new is the scale and complexity. As technology matures, our old solutions have started to break down. What worked for thousands or tens of thousands of files doesn't hold for hundreds of millions. This is a hard problem, and we're still learning. Our approach to recording and managing our data is just beginning and is just one path forward. I expect there are and will be others. I hope sharing what we've built sparks ideas, comparisons, and maybe even some collaboration. Let's wrap up all 3 of these talks. As you can see, We're so excited about MRD. It's poised to become one of Myriad's flagship tests, and we believe it will make an incredibly positive impact on our patients' lives. But the assay's complexity reflects its promise. We've encountered novel problems in both the science of the assay and the engineering required to support it. This is especially exciting for us. Novel problems are opportunities to forge ahead and discover novel solutions. It is both challenging and rewarding. Still, productionizing something like MRD is difficult. For the sake of our patients, we remain diligent and disciplined in our execution as we transform our research into a true product. Building MRD has been an enormous undertaking for many brilliant people across many teams and organizations. We want to make sure we acknowledge the many contributions made by the bioinformatics software team and our counterparts in the computational biology team. Strand Life Sciences has been instrumental. The assay development team, the LIMS team, Myriad's IT and SRE groups, the clinical development team, and the project management team, with additional heartfelt thanks to our many study partners. We'd love to answer your questions. Also, is there
A question here.
Hi, pretty nice talk. And in your workflow, I noticed that the alignment and the variant calling are separated out in two different platforms, and one is DRAGEN, one is the DNAnexus. So what is the specific reason to separate them out? Have you guys considered kind of use one platform to do both of the steps? Thank you.
If I understand the question is why we use two different platforms to perform alignment. Well, one of the alignments is for is for tumor profiling, and the other one is for ctDNA detection. The aligners that we use for both of them have their own advantages and disadvantages. We were able to get DRAGEN to a place that we were comfortable with using it in a tumor profiling assay, and we're just not there to as far as that goes for the detection part of our assay at the moment.
I see. So it's like a two different platform, like have specific use case, right?
Yeah, and like the specific details of why one aligner chosen over another is not part of our team,
Nice.
but there's other considerations as far as getting scientific validation, and we're validating right now using the setup that we have.
Thank you.
Sorry, I couldn't give a more detailed answer. Not really our specific area. The presenting team right here, right now.
Yeah, one more question.
Hi, I have a question for Benjamin about your concurrency issue. I was wondering if you had any learnings about how you would like preemptively stress test the system with production like levels of concurrency or scale to kind of like proactively identify any weaknesses as opposed to like having to react in the moment to like a crisis situation.
Yeah, that is a great point. There are, so for specifically the lambda concurrency where we had the concurrency of one. I don't know if that's the one you're talking about, or if you were talking about the other one, or just both of them. But definitely stress testing is on the list. Testing the orchestrator with large amounts of data is actually a little bit difficult to do because of the number of systems involved, and the number of synchronization between those systems. It is actually quite a difficult task. The one that we are looking into deeply, and it is something that is a definite goal of ours, is to be able to better test that. Absolutely.
To add to that, load and performance testing are some of the most difficult kinds of testing you can perform. What Benjamin observes is that like the design of the system that we have with MRD is not built with load testing in mind on the orchestrator side. And as a consequence, testing load there is difficult. If you're concerned about this particular problem and you have not yet designed your system, it may be worth taking a moment to think ahead about how you would inject a lot of work into the system in a testable fashion. It's been a while since I've worked in that problem space, and so I can't make any recommendations on tooling, but there's sure to be tooling available. The other thing to consider is to characterize the loads that you do expect ahead of time, so that you at least have an idea of what you want to design your system to support. But yeah, design your system for testability if you can. Otherwise, you're going to be in for a lot of headaches setting up your load testing harness.
Yeah, and this sort of also goes back to one of the slides I brought up earlier in this transition from R&D into production. That crisis, I don't know if we would call it quite that deep of a term, but that concurrency issue happened in R&D, and that was one of the moment that we realized that we had an issue with concurrency was at that moment. So, any other questions?
Another question here.
Yeah, I guess just I really like the talk. I like the different perspectives here. This is really great. But like it seemed like the concurrency issue and other issues were coming up largely because of the use of Lambda. And I guess I'm just kind of confused, like especially if this is supposed to be a production environment, why use AWS Lambda then? Like what I understand the general advantages of Lambda, but like I don't necessarily understand them here in this use case.
There are two sides to this question. So our state machines use lambdas as their step executors. The DMS uses lambdas as kind of event handlers, be they API events or the more traditional EDA side of things. The truth is, we may end up deciding in the case of these really big operations, like say, for instance, the getting a whole bunch of S3 events all at the same time when we make a big operation, like Benjamin found, where we ended up introducing the RDS proxy, where we ended up adding the SQSQ. It may end up being more appropriate to stand up something like a Fargate instance to handle these like big, maybe better serialized than parallelized operations. We haven't characterized that yet. This is all quite new in our space, and so we're learning as we go along. I don't know if that's a complete answer. On the step function side of things, it's worth pointing out that we really don't expect to be handling thousands and thousands and thousands of operations at once. Usually it's more of a state machine operation. The thundering herd problem, that's a different matter entirely. I don't know if that answers your question entirely.
Yeah, I would just like to add that, like, serverless, AWS Lambda, the state machines, these are all powerful tools. Powerful tools that can also allow you to shoot yourself in the foot with them, right? Like, some of these are architectural mistakes, right? Like we didn't set them up quite the way that they ought to have been, and we discovered them in this production environment, actually in R&D, most of these. And that is, part of this talk here is now to prevent you from making those same mistakes, and recognizing things like account concurrency are an important thing to keep an eye on, that events could quickly overload that account concurrency, that when you are designing a state machine and you need to set one of the concurrency levels to 1, that might be an indication that you need to think about why that is and think at a bigger level. Certainly. But I wouldn't say like it's a great tool, and I think we're going to continue to use it, but we're always reevaluating our tools.
Can I ask a question?
Yeah.
So you're mapping, well, you have 1,000 targets at the end from the somatic variant calling, right? The tumor normal subtraction.
Mm.
Have you considered just mapping to the targets? Like, I'm assuming you're mapping to the human genome with your subsequent alignments, but do you really need to?
Yeah.
Maybe this is a different team.
Yeah, I'm afraid it sounds like a great question and one that I would like to hear the answer to myself. But I don't have the context or background to give a good answer to that question. So I'm gonna perhaps if you write it down, maybe we can get you an answer. But yeah, I don't. We're not the team for that. Sorry.
Yeah, no problem. Thanks.
I had a question. The DMS policy, how is it described? Is there a language that describes it, or do you have to code it in?
In general, I am very suspicious of domain-specific languages, and so our intention is to describe them directly as code. We are still in the process of arriving at the design for this as a finalized version of it. I suspect we would prefer to lean on the interpreter. This is a Python-based project. We'd like to lean on the interpreter a bit when building out these policies. There is something to be said about like writing up a YAML or a TOML for this sort of thing. However, I suspect there will be complexity involved there that will be better encapsulated with code as opposed to especially a domain-specific language. Like SQL is powerful, but in general, building out a DSL can be really quite thorny.
Yeah, because you'll have to write your own parser, I guess. Yeah, I didn't think of that. Sorry.
Oh,
Yeah.
no, it's a good question.
Makes sense. Yeah. Thanks again to all the three speakers. Maybe we'll have a round again, please.